All right. A quiz for cannabinoid experts, huh?
Yeah.
You really threw us a curveball with this one.
Yeah.
I'm excited to get into this, though. Ready to unpack some serious cannabinoid science with me.
Absolutely. It's definitely a fascinating challenge. We've got the cannabinoid expert quiz that you found and a whole stack of research papers and texts to guide our deep dive. So yeah, listeners, get ready for a really in-depth graduate level exploration of these really intricate molecules.
Let's jump right into it. First question from the quiz. Which amino acid in the CB1 receptor is crucial for G protein sequestration and constitutive activity? Now, I know our listeners are pretty familiar with cannabinoid science, but let's break this down a bit. Why is this question so important?
Well, it really gets at the core of how the CB1 receptor functions. We're talking about those tiny molecular details that determine how cannabinoids like THC actually work, how they do their thing in the body. The quiz gives us the answer. It's aspartic acid at position 2.50, or if we're talking about the human CB1 receptor, position 163.
Okay, so aspartic acid 2.50 it is. But why is this one particular amino acid so important? It's just one tiny piece of a much larger protein, right?
Right, but that's what makes it so fascinating. This aspartic acid sits in a really, really highly conserved region of G protein-coupled receptors. It's called the DRY motif. You can think of this motif kind of like a molecular switch. It flips when the receptor gets activated.
So this aspartic acid is like part of the mechanism that allows the CB1 receptor to turn on.
Exactly. And its position within that DRY motif really makes us think that it's got something to do with those shape changes that happen, those conformational changes, when a cannabinoid, anything, binds to the receptor.
Okay, so what happens if this aspartic acid isn't doing its job right?
Yeah.
What if there's a mutation at that position? What happens then?
Well, Aboud's research showed that mutations there at that position can cause all kinds of interesting effects. Some mutations can totally abolish G protein sequestration, and that leads to something called constitutive activity. Basically, imagine the receptor is constantly sending signals even when nothing is bound to it.
So it's like the receptor stuck in the on position. That sounds like it could cause some problems.
Exactly. That constitutive activity, it can really mess with that careful balance in the endocannabinoid system. And that could mean unexpected side effects, even bad side effects, potentially.
Yeah, that makes sense. The research also talks about how these mutations can impact G protein coupling. Can you remind us what that means and why it's so important for cannabinoid signaling?
Sure. So G proteins, those are like little messengers, molecular messengers, I guess I should say. They relay signals from the receptor to other parts of the cell that starts a whole chain of events, like a domino effect. So if the receptor can't connect with those G proteins properly, that whole chain reaction, that whole signaling pathway gets disrupted.
So it's like cutting the communication lines between the receptor and the rest of the cell.
Yeah.
Right.
Exactly. That's a great way to put it. And when that communication breaks down, it can impact a bunch of fundamental processes in the cell. Everything from inhibiting adenylate cyclists, which Hallett and Fleming did a ton of research on, to modulating ion channels, all these really basic processes that control how our bodies work.
It's wild to think that one tiny change, just a single amino acid in this giant protein, can have such huge effects at a cellular level.
Yeah.
Okay, on to the next question from this expert quiz. The side chain of delta-8 THC is crucial for its activity. What structural feature of the side chain is particularly important for CB1 receptor affinity and potency?
So this one really gets into that structure-activity relationship of cannabinoids, It highlights how even tiny molecular tweaks can totally change their effects. The answer here is conformational freedom in the side chain.
Okay, conformational freedom. Break that down for us who aren't organic chemists.
Think about it like this. For a key to work in a lock, it needs to be the right shape, obviously, but it also needs to be able to move in a specific way. In the same way, for delta-8 THC to properly bind to that CB1 receptor, its side chain has to be able to adopt a specific conformation, a specific three-dimensional shape.
So if the side chain is too rigid, that'd be like a stiff key that can't turn in the lock.
Exactly. Thomas and his team actually did some cool research on this. They used computer modeling and something called QSR analysis to look at a whole bunch of delta-8 THC analogs. And they found that if you restrict the side chain's flexibility, it's like making that key stiff, you actually end up creating a CB1 receptor antagonist.
So instead of activating the receptor, it blocks it. That seems kind of counterintuitive. Wouldn't you want the cannabinoid to bind strongly and activate the receptor?
It seems that way at first, but it shows how complex these interactions really are. Blocking a receptor can be just as useful therapeutically as activating it sometimes. Razdan also found this in their work. They made some classic CB1 antagonists just by limiting the side chain's conformational freedom.
Wow, so it's all about finding the right balance. Right. It needs the right binding affinity and the ability to cause the right changes in the receptor shape. Too rigid, and it blocks the receptor. Too flexible, and it might not bind strongly enough.
Yeah, that's a great way to sum it up. It's this delicate dance of molecular interactions.
Okay, let's keep going. Next question from this expert-level quiz. What is the primary mechanism of action of the endocannabinoid anandamide in regulating human sperm function? Now we're getting into the endocannabinoid system's role in reproduction.
This is a fascinating area of research. So the answer is that anandamide mainly impacts sperm function by changing the properties of the sperm cell membranes.
Interesting. So we're not talking about direct hormonal effects or anything.
Not directly, no. The research points to anandamide's interaction with the sperm membranes as being key to how it affects things like capacitation.
Okay, quick refresher. Capacitation. What is that again, and why is it so important for fertilization?
Basically, capacitation is a bunch of biochemical changes that sperm have to go through in the female reproductive tract. It's how they become able to fertilize an egg. It involves changes in things like membrane fluidity, protein phosphorylation, and intracellular calcium levels.
So anandamide is affecting those membrane changes that are so important for sperm to actually do their job.
Exactly. Research suggests that anandamide and also its buddy, palmitoylethanolamide, can affect the timing and how capacitation happens. They do that by changing the fluidity and permeability of sperm membranes.
Are there any examples from the research that show this effect?
Oh yeah, for sure. One study found that anandamide at similar concentrations to what you'd find in human seminal plasma actually reduced the fertilizing capacity of sea urchin sperm. It actually inhibited the acrosome reaction.
Whoa, inhibiting fertilization. That's a pretty big deal.
Oh yeah, definitely. The acrosome reaction is a key step in fertilization. It's when the sperm releases enzymes that allow it to get through the egg's outer layer. This finding suggests that, at least at certain concentrations, an anandamide could actually work as a contraceptive.
That's wild. So the endocannabinoid system isn't just about regulating things like our mood, appetite, and pain. It's playing a part in, like, the very essence of life itself. Reproduction.
Exactly. It really shows how big and unexplored the influence of the endocannabinoid system is on our physiology. The research on anandamide and sperm is still ongoing, but these findings suggest it might be a key regulator of fertility, both in promoting and potentially inhibiting fertilization.
Okay, on to another question from this tough quiz. It asks, which cannabinoid has been shown to be particularly effective in the treatment of neuropathic pain, specifically in multiple sclerosis?
This question takes us to the realm of therapeutic applications. This is a field that's booming right now as research uncovers all the potential benefits of cannabinoids. The answer, based on a lot of clinical research and the quiz itself, is Sativex, a combination of THC and CBD.
Sativex, a name that a lot of people in the medical cannabis world will recognize. Now, you mentioned neuropathic pain. That can be really, really debilitating. For people who might not be familiar with it, can you explain what makes it unique?
Sure. Neuropathic pain comes from damage or problems within the nervous system itself. Imagine that the wires that carry pain signals are, like, frayed or misfiring. It's often described as a burning, shooting, or stabbing pain, and it's often super resistant to normal pain medications.
And why is Sativex particularly effective for this kind of pain, especially in MSK?
So, multiple sclerosis is an autoimmune disease that attacks the myelin sheath, the protective covering around nerve fibers. This causes all sorts of neurological problems, and one of them is neuropathic pain. Sativex seems to be good at managing this pain because of the way THC and CBD work together. They have synergistic actions.
Meaning they're better together than on their own, like a dynamic duo of pain relief.
Exactly. We know THC activates CB1 receptors and has analgesic effects, but CBD helps by reducing inflammation and maybe modulating other pain pathways that often go haywire in neuropathic pain.
So it's like a multi-pronged attack on the pain, hitting it from different angles. Are there studies that back up how effective Sativex is for this?
Oh yeah, tons. Lots of randomized, double-blind, placebo-controlled trials. The gold standard of clinical research show how well Sativex works for reducing neuropathic pain in people with MS. One study by Novotna and their team found big improvements in pain scores and spasticity in patients who had refractory spasticity caused by MS and who were given Sativex on top of their other treatments.
refractory spasticity, meaning it didn't respond to other treatments.
Yeah.
So for people who haven't found relief from the usual therapies, Sativex could be a real game changer.
Exactly. It shows how much potential cannabinoid-based therapies have for people who haven't found relief from other treatments. Research is still ongoing to figure out the long-term effects and safety of Sativex, but these early findings are really, really promising for people dealing with this really tough condition.
Let's move on to another intriguing question from this expert-level quiz. Abian CBD exhibits unique pharmacological properties, particularly in the cardiovascular system. What receptor does it appear to target, and what is its effect? This one brings up a less well-known cannabinoid.
Right, and it has some really interesting effects. So Abian CBD is a synthetic version of cannabidiol. It seems to target a completely new endothelial cannabinoid receptor. It's different from both CB1 and CB2.
Okay, an endothelial cannabinoid receptor, meaning it's found in the lining of blood vessels.
Exactly. And the really cool thing about Abian CBD is that even though it doesn't bind to CB1 or CB2, it still causes some big changes in the cardiovascular system. Specifically, it causes hypotension.
Hypotension, so a drop in blood pressure.
Yep, that's right. Jari and his colleagues really dug into this effect. They found that Abian CBD caused hypotension in both normal mice and mice that had their CB1 receptors knocked out. That means that its effects don't depend on the CB1 receptor.
So it's like it's bypassing that usual CB1 pathway we've been talking about. Wow, that's really interesting. Makes you think there's more to the cannabinoid system than we initially realized, huh?
For sure. More research suggested that Abian CBD's effect on blood pressure probably comes from the release of something called an endothelium-derived hyperpolarizing factor, or EDHF for short.
EDHF. Can you unpack that for us? How does that lower blood pressure?
So EDHFs are basically a group of signaling molecules that make blood vessels relax. This vasodilation leads to a decrease in blood pressure. Jari's study found that when they used inhibitors to block calcium-activated potassium channels, which are involved in that EDHF vasodilation, the vasodilatory effects of Abian CBD were significantly reduced.
So basically, Abian CBD triggers the release of these EDHFs, which then relax the blood vessels and lower blood pressure. And all of this happens without even touching CB1 or CB2 receptors.
That's it. Finding this new endothelial cannabinoid receptor and figuring out how Abian CBD works is really exciting for developing targeted cardiovascular therapies. It just shows how much we still have to learn about cannabinoid pharmacology. We've only just started to scratch the surface of this system and all its potential.
All right, one more question from this expert-level quiz before we wrap up this first part of our deep dives. Which cannabinoid has shown potential as an anxiolytic agent in human studies, and what brain areas are believed to mediate this effect?
This question is all about how cannabinoids might be used to treat anxiety, which affects so many people around the world. The answer, based on the quiz and the research out there, is CBD.
CBD, always popping up with new potential uses. This molecule really seems to be a jack of all trades when it comes to cannabinoids.
It really is so versatile. And unlike THC, which can actually cause anxiety in some people, CBD has shown calming and anxiolytic effects in human studies.
So if THC is like the party animal, CBD is like the chill, relaxed sibling.
That's a great way to put it. Zordi and his team did some really interesting research on CBD and anxiety. They found that a 300 milligram dose of CBD significantly reduced anxiety that was brought on by a simulated public speaking test. Other studies have found similar anxiolytic and sedative effects of CBD in healthy people, too.
Public speaking, definitely a good way to make most people anxious. What about the brain areas that are involved in these calming effects? How does CBD actually work to reduce anxiety?
Research suggests that the limbic and paralimbic brain regions are key here. Those are the areas that deal with processing emotions, memories, and fear and anxiety-related behaviors.
So CBD is working in the very parts of the brain that control our anxiety response.
Mhm.
That's pretty remarkable.
It is, and get this. Research suggests that CBD can actually counteract the anxiety-inducing effects of THC in some people.
So it's like a built-in anxiety buffer.
You could say that. It highlights the complex and often opposing effects that different cannabinoids can have within the endocannabinoid system. They interact with each other, with other neurotransmitter systems, and with different brain regions to fine-tune our emotional responses and overall well-being.
Wow. So there we have it. We've tackled some really tough questions and gotten into the nitty-gritty of cannabinoid science.
Welcome back to our deep dive into all things cannabinoids. We're really digging into the mechanisms and potential therapeutic uses of these complex molecules. And you know, your expert quiz has been a fantastic guide for us.
Yeah, it's been like a real scientific expedition, uncovering the secrets of the endocannabinoid system one question at a time.
Exactly. Each question is a new path, right? Leading us to a better understanding of this complex system and what it does in the human body.
All right, let's tackle the next question then. This one asks, how does chronic administration of delta-9 THC affect testosterone secretion and sperm production in animal models? Now we're getting into some interesting, maybe even controversial territory, talking about cannabis and male reproductive health.
For sure. This question has definitely sparked a ton of research, and the findings are really interesting. Chronic exposure to delta 9 THC in animal models seems to have a negative impact on both testosterone production and sperm parameters.
So regular cannabis use could potentially mess with a guy's fertility. That's a pretty big deal.
Yeah, it is. And researchers have been looking into this for a while. Studies have shown that when you give delta 9 THC to animals over a long period of time, it can lead to lower testosterone levels. And that's probably because of how it affects the hypothalamic pituitary gonadal axis, the hormonal control center for reproduction.
So basically, THC is messing with the hormones that regulate testosterone production.
Exactly. And when those hormones get disrupted, it can lead to less sperm production, less sperm motility, and lower sperm viability. Basically, a whole cascade of effects that could end up impacting a guy's ability to have kids.
Are there specific examples from the research that really show these effects?
Oh, absolutely. Macaron and his team did a study that dug deep into how the endocannabinoid system is involved in spermatogenesis. They focused specifically on how anandamide affects Sertoli cells.
Sertoli cells, those are the ones that support and nourish the developing sperm, right? Like they're the nannies of the testes, making sure those sperm cells grow up big and strong.
Haha, that's a great way to put it. They're totally crucial for spermatogenesis, that whole process of sperm development. So this study found that if you give those Sertoli cells high concentrations of anandamide, it can actually cause apoptosis, basically programmed cell death.
So too much anandamide could lead to the death of the very cells that are essential for sperm development. That's not good.
Yeah, it's a bit of a concerning finding, for sure. But it's important to remember that they saw this effect at high concentrations of anandamide. We need more research to understand how chronic THC use affects endocannabinoid levels in the testes, and also what the long-term effects are on human male fertility.
Definitely. It's a good reminder that while cannabinoids have so much therapeutic potential, there can also be risks. It's all about finding that sweet spot, right? Getting the benefits and minimizing the potential downsides. Okay, let's move on to the next question. What is the role of the C11 position in determining the pharmacological activity of delta-9 THC? Back to the molecular details.
Back to the nitty-gritty. Yeah, this question is all about the structure-activity relationship of THC and how it interacts with the CB1 receptor. That C11 position is really important for how potent and active THC is.
So if we mess with that specific spot on the molecule, we can change how it interacts with the CB1 receptor, like adjusting the settings on a machine to change how it works.
You got it. Research has shown that even small changes at the C11 position can dramatically affect THC's effects.
Can you give us some examples, like what kinds of changes and what do they do?
Sure, let's start with hydroxylation. Adding a hydroxyl group to the C11 position, like an 11-hydroxy-THC, actually makes the molecule more potent. That hydroxyl group helps it interact with the CB1 receptor better, making it bind more tightly.
So it's like making the key stickier so it grabs onto the lock better.
Exactly. But on the other hand, if you get rid of the C11 hydroxyl group or swap it out for something bulky, it usually makes THC much less active. It's like trying to jam a giant key with a huge keychain into a tiny lock. Not going to work.
So it's all about finding that just right size at the C11 position. Not too big, not too small, just right to get the best effects from THC. Have researchers tried other tweaks at that position?
Oh yeah, they tried all sorts of things. Adding alkyl groups, attaching amino-alkyl chains, all kinds of modifications. For example, the N-methyl analog of THC, that one turned out to be just as potent as delta-9 THC in depressing spontaneous activity in mice. But adding bigger, bulkier groups to that C11 spot generally made the molecule much less active.
It's crazy how such tiny changes can make such big differences in how these compounds work. Okay, let's shift gears a bit. This next question gets into how the endocannabinoid system affects our thinking. Blocking CB1 receptors has been shown to enhance memory duration in some animal models. What is a possible explanation for this effect? So now we're talking about learning and memory.
Right. This is where the endocannabinoid system gets really interesting. The answer lies in that delicate balance between how active neurons are and synaptic plasticity, the brain's ability to change and adapt based on experiences.
Synaptic plasticity, that's how our brains learn and form memories, right? It's like the brain rewiring itself based on what we go through.
Exactly. And the endocannabinoid system plays a complex role in controlling that plasticity. Blocking CB1 receptors can actually tilt things in favor of memory formation.
Okay, but how does that actually work? How does blocking those receptors lead to better memory? Doesn't seem like it should, really.
Well, one idea is that endocannabinoids actually inhibit certain parts of the brain that are involved in memory,
Hmm.
specifically the hippocampus, which is like the memory center of the brain. When CB1 receptors are activated, they stop neurotransmitters from being released. That can quiet down neuronal activity and limit synaptic plasticity.
So by blocking the CB1 receptors, we're taking off the brakes on those memory parts of the brain, letting them be more active and form stronger connections and memories.
Exactly. That increased activity can then make long-term potentiation, or LTP, happen more easily. And LTP is a really important mechanism for making memories.
LTP, that's like strengthening the connections between neurons, right?
Yeah.
The stronger those connections, the stronger the memory.
That's it. The research mentions a study by Varvel and Lickman. They were looking at the endocannabinoid system's role in learning and memory, and they found that blocking CB1 receptors with a specific antagonist called SR1 41316 actually made memories last longer in a bunch of different animal models of learning and memory.
So that antagonist basically shuts down those CV1 receptors. And then those memory parts of the brain can fire up and make those stronger connections. It's like taking the brain off autopilot so it can learn better, right?
Haha, I like that analogy. Of course, this is just one possible explanation. We need more research to really understand the complex mechanisms at work here, but it shows how much potential CD1 antagonists might have as cognitive enhancers. Definitely an area for future research.
Definitely. Okay, let's switch gears again and talk about THC's effects on the heart and blood vessels. The quiz asks, what role do prostanoids play in mediating the cardiovascular effects of THC?
This question reminds us that different systems in the body are all connected, and THC doesn't just affect the brain, it can impact cardiovascular health too. Prostanoids are a group of lipid signaling molecules. They're key players in both the vasodilatory and vasoconstrictive effects of THC.
Okay, sensing a theme here. THC seems to be good at producing opposite effects depending on the situation. It's like a molecular chameleon blending in wherever it goes.
Haha, yeah. It's definitely complex. The cardiovascular effects of THC are complicated. They can depend on how much you take, how you take it, and just individual differences in people's physiology.
So how do these prostanoids fit into all this? How are they involved in that push and pull between vasodilation and vasoconstriction?
Well, research shows that THC can actually stimulate the production of different prostanoids, including prostacyclin and thromboxane A2. Prostacyclin, which is made by the cells that line blood vessels, is a really strong vasodilator. It relaxes the walls of blood vessels, which lowers blood pressure.
So THC is promoting vasodilation, in this case, by increasing prostacyclin. What about thromboxane A2? Does that also help with relaxation?
Nope, not quite. Thromboxane A2, which is mostly made by platelets, actually has the opposite effect. It's a vasoconstrictor, meaning it makes blood vessels constrict. That can lead to an increase in blood pressure.
So THC can either lower or raise blood pressure depending on which prostanoid pathway gets activated, like having two volume knobs on a stereo, one to turn it up and one to turn it down.
That's a great way to put it. The balance between these opposite effects can be different for everyone. It depends on individual factors and the specific circumstances.
Definitely a delicate balancing act. It just highlights how important it is to understand individual responses to cannabinoids, since they can be so different for different people. Okay, let's move on to a question about CBD's anti-inflammatory potential. What evidence suggests that CBD may have anti-inflammatory properties?
This is one of the hottest areas of CBD research, and for good reason. There's a ton of evidence, both from lab studies and animal studies, that shows CBD's anti-inflammatory effects.
So CBD is like, what, a molecular firefighter? Calming down those inflammatory fires in the body?
Haha, I like that. That's a good one. CBD can actually modulate a bunch of key pathways that are involved in inflammation, things like the production of cytokines, chemokines, and pro-inflammatory enzymes. Those are the signals that drive the inflammatory response. And CBD can basically tell them to chill out.
Cytokines, chemokines, remind us what those are and why they're so important in inflammation.
Think of them like the communication system for inflammation. Cytokines are proteins that tell immune cells what to do, including how to create inflammation. And chemokines, those are a specific type of cytokine that call immune cells to the spot where inflammation is happening.
So CBD is basically messing with the immune system's communication network, telling those inflammatory signals to back off, like a peace treaty between the immune system and the body.
Exactly. CBD can actually reduce the production of those pro-inflammatory cytokines, things like TNF-alpha and IL-6, which are major players in inflammation.
Gotcha. Are there any studies that really show CBD's anti-inflammatory effects in action?
Tons of them. One study by Ribeiro and his team looked at how CBD affected acute lung injury. They triggered the injury using LPS, which is a strong inflammatory trigger. And they found that pre-treating with CBD actually significantly reduced how many neutrophils went to the lungs. It also decreased how much albumin leaked into the airways. And it suppressed the production of those inflammatory signals, TNF-alpha and IL-6.
Okay, neutrophils, albumin leakage. Break that down for us non-medical folks.
Right.
What do those things actually mean in terms of what's happening in the lungs?
Sure. So neutrophils are a type of white blood cell. They're the first responders to injury or infection. They rush in to fight off invaders. But if too many get activated, they can actually damage tissue. Albumin is a protein that's normally just in the blood. If it starts leaking into the airways, that means the lungs are inflamed and getting more permeable.
So CBD was able to calm down that whole inflammatory cascade in the lungs, right? Fewer neutrophils, less leakage, and fewer inflammatory signals. It's like calming a storm in there.
Yep, exactly. Another study by Li and their colleagues looked at how CBD affected acute pancreatitis, which is a serious inflammatory condition of the pancreas. They found that CBD actually improved how the pancreas looked, and it also decreased those inflammatory markers, IL-6 and TNF-alpha.
So CBD was helpful in different organs and different inflammatory conditions. Sounds like it could be a valuable tool for a lot of different inflammatory diseases.
That's what the research suggests. We still need more research to figure out the full potential of CBD, but these findings are really promising for people who are looking for relief from inflammatory conditions.
For sure. Okay, let's shift gears again. This next question asks, what are some of the challenges in developing cannabis-based medicines, and what are some strategies to overcome them? Now we're talking about the actual development of pharmaceuticals.
Right. Bringing cannabis-based medicines to market is a long and complicated process. It's got challenges that go beyond the typical hurdles of drug development.
What makes cannabis-based medicines so tricky to develop? What are those unique challenges that researchers and companies face?
Well, there are a bunch of things that make it tough. First, cannabis itself is really variable. As a plant, its chemical makeup can change a lot depending on the strain, how it was grown, and how it was processed. This makes it hard to standardize doses and make sure the effects are consistent.
So it's like trying to make a precise drug from a plant that's all over the place. Each batch could be a little different, which would make it tough to guarantee consistent results.
Yeah, exactly. Another challenge is the psychoactive effects of THC. THC definitely has some therapeutic benefits, but it can also make people feel high and impair their thinking, and that raises concerns about safety and the potential for abuse.
Yeah, you don't want a medication that makes you high when you're trying to treat pain or anxiety. That defeats the purpose.
For sure. And then there's the stigma around cannabis, which can make it hard to get research funding and regulatory approval. And of course, the legal status of cannabis is a whole other mess, since it varies so much from country to country and even within countries.
It's like trying to navigate a minefield of laws and regulations just to get a cannabis-based medicine approved. So with all these challenges, what are some ways to overcome them? How can we harness the benefits of cannabis and address these hurdles at the same time?
One important strategy is to focus on making standardized extracts or isolating specific cannabinoids. That way, you have a well-defined chemical profile and can control the dose more precisely. This helps to ensure consistency from batch to batch.
So moving away from the whole plant and towards specific purified compounds, like extracting the essential ingredients from a complex recipe.
Exactly. Sativex, which we talked about earlier, is a good example of this. It's a standardized spray with a set ratio of THC and CBD. That makes the dosing consistent and the effects more predictable. Another strategy is to look for new ways to deliver the drugs, ways that can improve bioavailability and minimize those psychoactive effects.
Bioavailability, meaning how much of the drug actually gets into the bloodstream and to where it needs to go.
Right.
Right. It's like making sure the medicine actually reaches its destination without getting lost on the way.
Right. For example, things like transdermal patches or buccal films can release cannabinoids slowly and in a controlled way. That way they bypass the liver, which can break down a lot of the drug, and that can potentially reduce those psychoactive effects.
Interesting. So finding clever ways to deliver those helpful cannabinoids while minimizing the downsides. What about the stigma and the legal hurdles? How do you deal with those?
Tackling the stigma takes a multi-pronged approach. We need more research that shows how effective cannabinoids can be. That will help change public opinion. We also need to be open and honest about both the risks and benefits of cannabis-based medicines.
Education and transparency are key. We need to separate the science from the stigma so people can make informed choices based on evidence instead of fear or misinformation. What about the regulatory side? How can we deal with that?
We need to work together with regulatory agencies to set clear rules and standards for developing and approving cannabis-based medicines. That means running rigorous clinical trials to prove they're safe and effective. It also means having strong quality control measures.
So it's a long and difficult process. But with the right strategies and partnerships, it seems like we can overcome these obstacles. It takes perseverance, creativity, and a real commitment to bringing these potentially life-changing treatments to people who need them.
Okay, let's look at a question that focuses on what patients report during clinical trials. Describe a patient-reported outcome measure that has been used in clinical trials to assess the effectiveness of Sativex in treating spasticity in multiple sclerosis.
This is a really important point. We need to understand the patient's experience when we're evaluating how well a treatment works, especially for symptoms like spasticity, which are subjective. It's not just about the numbers, it's about how the treatment is affecting the patient's quality of life.
Right. The patient's perspective is crucial. We need to know how a treatment impacts their everyday life, their ability to move, function, and just feel comfortable.
Absolutely. For Sativex trials in MS, the numeric rating scale, or NRS, has been used a lot to measure patient-reported outcomes. Okay, the NRS. What is that, and how does it work? How does it capture what the patient is feeling with spasticity?
The NRS is a simple scale that asks patients to rate how severe their symptoms are, in this case, their spasticity. They rate it from 0 to 10, where 0 means no spasticity at all, and 10 is the worst imaginable spasticity.
So it's like a pain scale, but for spasticity, a way for patients to describe how it feels, even though it's subjective.
Yeah, exactly. The NRS lets patients quantify their experience. It gives us data on how their symptoms are changing over time as they use the treatment.
Simple, but effective. Are there examples from the research that show how the NRS has been used in Sativex trials?
Oh, for sure. The source material mentions that Novotna and their team, in the study we talked about before, used the NRS to track how well Sativex was working for patients with refractory spasticity caused by NRS. They found that patients who took Sativex had significant reductions in their NRS scores. That means their spasticity was actually getting better according to the patients themselves.
So the patients were actually feeling a real improvement in their spasticity thanks to Sativex. It's not just about numbers on a chart, it's about real changes in people's lives.
Absolutely. The source material also mentions a review by Sneed and their colleagues. They emphasized how important these patient-reported measures are for capturing the functional impact of spasticity. It's not just about lowering a number on a scale. It's about how that translates into the patient's daily life. Can they walk better, dress themselves, do the things they enjoy?
Yeah, that makes sense. It's about restoring function, making life better, and giving patients back some control over their bodies. Okay, let's dive into a question about the endocannabinoid system and how it works with other neurotransmitter systems that control our appetite. How does the endocannabinoid system interact with other neurotransmitter systems involved in appetite regulation, such as the melanocortin and opioid systems?
This question takes us into that complex world of neurochemicals that control our appetite. The endocannabinoid system doesn't work alone. It's constantly interacting with other systems, like the melanocortin and opioid systems, to fine-tune our desire for food and our energy balance.
So it's like an orchestra, with different instruments playing their parts to regulate our appetite. Each system contributes to the overall feeling of hunger or fullness.
That's a great analogy. The melanocortin system, especially the melanocortin-4 receptor, or MC4R, is really important for suppressing appetite. When it's activated, it tells the brain we're full and we should stop eating.
So it's like the brakes on the appetite system. Where does the endocannabinoid system fit in? Does it help to press the brakes or ease up on them?
Well, research suggests that activating CB1 receptors can actually dampen the activity of the melanocortin system. This means that cannabinoids, especially THC, can increase appetite by weakening those inhibitory signals from the MC4R.
Ah, so THC is making those brakes less effective, letting us keep eating even when we're already full. explains the munchies.
Ah, exactly. There's been a lot of research on this interaction between the endocannabinoid and melanocortin systems. Verdi and colleagues did a study where they found that a CB1 antagonist called rimonabant actually made the appetite-suppressing effects of alpha-melanocyte-stimulating hormones stronger, and that hormone activates the MC4R.
So blocking those CB1 receptors with rimonabant allowed the melanocortin system to do its job better, putting the brakes back on appetite.
Right. Now let's talk about the opioid system. Opioids, like endorphins, are known to stimulate appetite, especially for tasty, rewarding foods.
So opioids are like hitting the gas pedal on appetite, making us want those delicious treats. Do they work together with the endocannabinoid system to make those cravings even stronger?
It seems that way. Research suggests that the endocannabinoid and opioid systems actually work together to make eating more rewarding. Activating both systems can boost dopamine release in the brain's reward centers, which makes eating more pleasurable.
So it's like a double whammy of pleasure when we eat. No wonder those cravings were so hard to resist when both systems are going strong.
For sure. This interaction might explain why certain foods, especially those that are high in fat and sugar, are so addictive. They take control of our reward systems, making us want them even when we're not truly hungry.
It's like our biology is working against us, making it hard to resist those temptations. Okay, let's wrap up this segment with a question about CBD's versatility. What are some of the potential therapeutic applications of CBD that are independent of its actions on CB1 and CB2 receptors?
This question reminds us that CBD does a lot more than just interact with those classic cannabinoid receptors. Even though CBD doesn't bind strongly to CD1 or CB2, it still has a wide range of effects that make it therapeutically promising.
So even though it's not directly activating those receptors, it's still doing good things in the body. It's like a behind-the-scenes operator working quietly to keep us healthy.
That's a great way to put it. One of CBD's best-known properties is its strong antioxidant activity.
Antioxidant, meaning it protects our cells from damage caused by free radicals, right? Those unstable molecules that cause aging and all sorts of diseases.
Exactly. Free radicals are like little troublemakers damaging DNA, proteins, and cell membranes. CBD can actually neutralize them, protecting cells from oxidative stress.
So CBD is like a bodyguard for our cells, fending off those free radical attacks.
I like it. And research shows that CBD is just as good, if not better, at fighting free radicals than classic antioxidants like vitamin C and vitamin E.
Wow, that's impressive. What else can CBD do that doesn't involve CB1 or CB2? Any other ways it's helping out?
Oh yeah, definitely. CBD can affect different signaling pathways and interact with other receptors in the body. That's one of the reasons it's so versatile. For example, it interacts with the serotonin 5-HT1A receptor, which is involved in mood, anxiety, and sleep. This interaction might explain why CBD can help with anxiety and depression.
So CBD is working on multiple fronts, influencing different neurotransmitter systems to produce all kinds of benefits. A real multitasker.
Absolutely. Another exciting area of research is how CBD might activate PPAR gamma receptors.
PPAR gamma, those are those receptors in the nucleus that control which genes are turned on and off, right?
That's right. Activating PPAR gamma has been linked to anti-inflammatory effects, insulin sensitization, and even neuroprotection. We need more research to fully understand how CBD works with these receptors, but it opens up some really cool possibilities for treating things like diabetes, metabolic syndrome, and neurodegenerative diseases.
Wow. That's a lot of potential for a molecule that doesn't even bind strongly to those main cannabinoid receptors, like a hidden talent just waiting to be explored.
Exactly. It just goes to show that we've only just begun to understand the vast therapeutic potential of CBD. It's a fascinating molecule with a bright future in medicine.
Welcome back to our deep dive into the world of cannabinoids. It's been quite a journey so far, right? Exploring the endocannabinoid system and the potential of these molecules.
Yeah, we've covered a lot of ground, from how they work at a molecular level to how we develop drugs and the challenges of taking what we learn from animals and applying it to humans.
And we've used this expert-level quiz as our guide, which has really pushed us to dig deep and explore the cutting edge of cannabinoid research.
It's been a fun challenge, and, it really highlights how much we still don't know about the endocannabinoid system. There's still so much to learn about its potential.
For sure. Okay, let's look at another question from this quiz. This one really takes us into uncharted territory. What is the evidence for the existence of non-CB1, non-CB2 cannabinoid receptors, and what are some potential candidates? Now we're going beyond those classic cannabinoid receptors.
Right. This question takes us to the edge of cannabinoid research, where scientists are looking for new receptors beyond CB1 and CB2. The evidence is still coming together, but we've got some interesting candidates that suggest the endocannabinoid system might be even more complex than we thought.
So it's like we've mapped out the major highways of the cannabinoid system, and now we're venturing off-road, looking for those hidden paths that could lead to new discoveries.
Exactly. One of the strongest pieces of evidence for these other receptors comes from studies that show cannabinoids still have effects even when you block both CB1 and CB2.
So if it's not CB1 or CB2 doing the work, those cannabinoids must be binding to something else, right? Some other receptor that's causing those effects.
Right. It suggests there are alternative receptors out there that we just don't fully understand yet. One possible candidate is GPR55. It's a G protein coupled receptor, but we don't know its exact function yet. It responds to some cannabinoids, including lysophosphatidylinositol or LPI.
GPR55, okay, another acronym for the list. So what do we know about what it might be doing in the endocannabinoid system? How does it fit into all of this?
Research on GPR55 is still pretty early, but studies have connected it to things like bone remodeling, pain perception, and inflammation. Henstridge and colleagues, for example, found that LPI, which might be a natural activator of GPR55, actually promoted calcium signaling and activated something called NFT, which suggests it's involved in cellular communication.
So even though GPR55 isn't a classic cannabinoid receptor, it seems to be involved in some of the same pathways. Like it's speaking the same language as the endocannabinoid system, but with a different accent. What other non-CB1, non-CB2 candidates are out there?
Another interesting one is the transient receptor potential vanilloid 1 receptor, or TRPV1 for short.
TRPV1, that sounds familiar.
Isn't that the receptor involved in feeling heat and pain? The one that capsaicin activates, the stuff that makes chili peppers hot.
Yep, that's the one. It's also involved in inflammation and other stuff, and research shows that some cannabinoids, CBD in particular, can actually interact with TRPV1. That might be how it affects pain and inflammation.
So CBD might be working through the same pathways that give us that spicy kick from chili peppers. That's a cool connection. It is. It shows how interconnected all these systems are and how cannabinoids can have all these unexpected effects. Another potential player is the peroxisome proliferator activated receptor alpha, or PPAR alpha. This one regulates fat metabolism and inflammation. And some cannabinoids, especially the ones with fatty acid side chains, can actually activate PPAR alpha.
Yeah.
So they might have therapeutic potential for metabolic disorders.
Wow, it's like these cannabinoids are master keys, opening all sorts of doors in the body, affecting so many different processes through these different receptors and signaling pathways. The endocannabinoid system is amazing, really.
Definitely. And the search for these non-CB1, non-CB2 receptors is ongoing. It's really exciting research that could uncover new therapeutic targets and help us understand just how complex the endocannabinoid system really is.
Speaking of research, this next question focuses on using animals in research. What are some of the limitations of using animal models to study the effects of cannabinoids on human health? Now we're talking about translating those lab findings into real-world applications.
This is a super important point. Animal models are really helpful for understanding how things work and finding potential new treatments, but they also have some limitations when we try to apply what we learn to humans.
So what works for a mouse might not work for a person.
Exactly. A lot of things can contribute to those differences. First, there are just basic differences in physiology and metabolism between species. A dose that's safe and effective for a rat might not be safe or have the same effects in a human.
Like trying to fit a square peg into a round hole.
Yeah, exactly. Another thing is that brains are different across species. Animal models can help us understand the brain circuits involved in behavior and disease, but the human brain is so much more complex. Small differences can change how cannabinoids affect things like thinking, mood, and behavior.
Yeah, the human brain is incredibly complex compared to the brains of most lab animals.
Yeah.
What other limitations should we be aware of when we look at research done on animals?
It's also hard to model those subjective experiences that humans have, Things like consciousness, emotions, and the influence of social and environmental factors. We can see how animals act, but we can't really know what they're feeling inside.
It's like trying to understand someone speaking a different language without a translator. You can hear the words, but you don't really get the meaning.
Exactly. And then there are ethical considerations, too. Some studies that might be important for understanding human health just aren't ethical to do in animals. We need to treat our animal subjects humanely, and that means setting limits.
So it's about finding a balance between pushing science forward and being ethical. So given these limitations, how can we make the most of animal models in cannabinoid research? How do we make sure that what we learn is relevant and applicable to humans?
It's important to choose the right animal model, one that's relevant to the specific research question. For example, some mice have been bred to have certain behaviors or conditions that are similar to human conditions, like anxiety or epilepsy. So you can study cannabinoids in a model that more closely reflects the human experience.
So it's about finding the right tool for the job.
Yeah. Another good strategy is to use multiple animal models. That way you can compare the results and make sure your data is solid. If you see the same effect across different species, it's more likely that it'll apply to humans too.
Like getting multiple perspectives to make sure you have the whole picture.
Right. And in the end, the goal is to take what we learn and test it in human clinical trials. That's where we can really see if cannabinoids work as therapies. Combining animal studies with lab experiments and human clinical trials gives us the best understanding of how cannabinoids affect human health.
Like putting together the pieces of a puzzle using different sources of information to get a complete picture.
I like that. We have to recognize the limitations of animal models and use a bunch of different research methods to move cannabinoid science forward and develop safe and effective treatments.
Okay, let's finish this deep dive with a look toward the future. This last question asks, what are some of the future directions and unanswered questions in cannabinoid research? We've explored what we know, but what's next? What mysteries are still waiting to be solved?
This is a great question. Cannabinoid research is moving so fast, and there's still so much we don't know.
So we've only just started to explore this whole world.
Totally. One of the biggest unanswered questions is just how big of a role the endocannabinoid system plays in our health and in diseases. We know the key players, the receptors, endocannabinoids, enzymes, but we're still figuring out how they all work together.
It's like we have the parts list, but we're still putting the machine together.
Yeah. And another big question is about the potential uses of cannabinoids for things other than pain and spasticity. We talked about CBD and its anti-inflammatory and anxiolytic properties, but researchers are looking at its potential in neurodegenerative diseases, cancer, and metabolic disorders too. There's so much potential there.
Feels like every day we hear about a new potential use for CBD.
Right. It's a really versatile molecule. Another exciting direction is developing new cannabinoid therapies that are more targeted, therapies that focus on specific receptors or pathways. That way we can maximize the benefits and minimize side effects. This means finding new molecules, but also better ways to deliver the drugs and maybe combining different therapies.
So it's not just about finding new drugs, it's about using them more effectively.
Exactly. And of course, we still have to deal with the stigma and legal issues around cannabis. Those things are holding back research and preventing patients from getting treatments that could really help them. We need to advocate for policies that are based on evidence and have honest conversations about the risks and benefits of cannabinoids. That's how we move the field forward.
It's a call to action for all of us, really. Scientists, policymakers, and the public. We need to work together to break down those barriers.
Absolutely. The future of cannabinoid research is incredibly exciting, and it has the potential to make a huge difference in people's lives. But to get there, we need to collaborate, be open to new ideas, and stay committed to doing good science ethically.
Well said. We've covered a lot in this deep dive, from the details of how cannabinoids work to their potential uses and the challenges we face. Hopefully you learned a lot and maybe even have some new questions to explore.
It's been a pleasure joining you on this journey. It seems like the more we learn, the more we realize how much more there is to discover. The endocannabinoid system is vast and full of potential, and it's exciting to think about what the future holds.
Keep asking questions and keep exploring.
Yeah.
Until next time, keep diving deep.